Good morning, good afternoon, and good evening, everyone. And welcome to this Akoya Biosciences and Scientist.com webinar titled Spatial Biology and the Gastrointestinal Tract, Unveiling Cellular Landscapes and Disease Mechanisms. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. John Hickey, who's Assistant Professor of Biomedical Engineering at Duke University, who will discuss how spatial biology is reshaping our understanding of the gastrointestinal tract from cellular diversity to disease mechanisms and treatment insights. And with that, I'm very pleased to welcome John Hickey. John, thanks so much for joining us today, and the floor is yours whenever you're ready.
Yeah, thanks for the introduction. This is exciting to be able to tell you about some of the work that we've been doing in understanding our digestive tract and using the Codex multiplex imaging platform from Akoya Biosciences. So I recently started my own lab at Duke University, but a lot of the work I will tell you about was done during my postdoctoral efforts. And done with a number of groups who I will acknowledge throughout and at the end. And today what I'll share is 3 projects. One on how we can use these spatial technologies to map healthy tissue and create cell atlases. And this is around the healthy human intestine. Another where we're profiling the progression of disease in chronic inflamed cancers like Barrett's esophagus. And then another project where we're looking at identifying specific macrophage niches, both within healthy and cancerous tissue. So just first to start out, I think it's really important to contextualize or define our cells within the context of these multi-scale interactions that they exist in. And we can take any tissue, this could be the intestine or cancer, for instance, and each tissue is made up of these multifunctional tissue units that are organized spatially, and they're interacting in localized ways to accomplish a larger function of the overall tissue. And then within these tissue units, you have distinct cellular neighborhoods, which are distinct compositions of cells, also spatially co-organized. And within these cellular neighborhoods, you have individual cells that are secreting proteins and ligands and interacting at a cell-to-cell level. And then within each individual cell, you have networks of molecular interactions. And, I think we've done a really good job of measuring these independently of each other. But I think to understand how biology is connected across these scales is really important to understand what are molecular drivers of tissue organization, but also why these break down in times of disease. So one of these methods that has been developed is Codex multiplex imaging, and this allows 50 plus molecular measurements at once within our tissue sections. From that, we can computationally define individual cell types, and from that, we can identify multicellular neighborhoods or structures. And then because we have all the data, we can then connect these across biological scales to gain insight. So in Gary Nolan's group, we developed this Codex multiplex imaging assay where you stain with 50 plus antibodies at once with DNA barcodes. You stain with as many antibodies as you have barcodes. This allows you to stain many more than you have fluorescent channels because we only use a complementary oligonucleotide attached to a fluorophore to image each of the individual antibodies. We strip off 3 at a time and then put on another 3 to image the net set until we've imaged them all. And so we've helped develop some of these protocols. How do you think about data storage and processing for such large imaging data sets? The types of metadata that should be standardized and along all of these different steps, from array creation to panel design to the actual imaging. How do you use, validate, and order these large antibody panels? Once you have the data, how do you think about in quantification and normalization and identifying individual cell types? And how do you make this information more machine readable? And so I'm not going to talk a lot about these protocols, methods, or techniques today. but more the application of all of this as a foundation. And one of the stories, like I mentioned before, is we wanted to understand how the human intestine is organized across different areas of the intestine. So starting from the duodenum and going all the way until we reach the sigmoid colon. And so we had the opportunity to take 4 different sites from the small intestine and 4 different sites from the large intestine. And these were from healthy donors. And we did codex multiplex imaging on each of these different areas, as well as single-cell RNA-seq and single-cell attack-seq. And this is what some of this imaging might look like, where we have 5 of the markers overlaid. And this magenta area is where we have our epithelial layer, which is next to the lumen of the intestine where your food is digested. And you can see lots of immune cells as marked in by CD3 or CD45 within these areas. Below, that's the mucosal area. Below this, you have the submucosa area where you have stromal, such as a vasculature or smooth muscle in the muscle layer, or things like an immune follicle. And because this has such great resolution, we can get down to single cells. Each individual little circle here is an individual cell. We can study things like the resolution or the development of immune follicles within tissues. We can look at cells in between these epithelial areas within the mucosa. We can also start to look at the stem cell niche of the intestine. So you can see we can plot many different markers and start to look at a lot of different biology. And the pictures are very pretty, but we want to go more beyond just creating the pictures. And so what we usually do at this stage is apply deep learning methods that do cell segmentation. And there's many different approaches. Essentially what this does is it outlines each individual cell with a mask, and that allows us to quantify the protein expression or the target molecule expression within each individual cell. And we use that much the same way that we would for CyTOF or single-cell RNA-seq, an unsupervised clustering method, where we identify individual cell types that are enriched for specific markers. The thing that's different about multiplex imaging is that sometimes there's additional noise in background, or cell segmentation is not perfect. And so this identification of data of cell types is a little more tricky. And this can take up to 20 hours a sample if you want high level accuracy annotations for all your data sets. And so I was reading and saw a cool paper by some computer scientists at Stanford that were working on classifying images. And one of the algorithms they were developing also was able to not only classify images in the training set, but it could classify new categories. And so we reached out to them, and we developed this algorithm called Cellular, which essentially takes in this multiplex spatially defined cell data set, where we've already labeled some of our spatial imaging data with the cell type labels as a tissue graph. Basically, each node in this graph represents an individual cell type, and then it is connected via edges to other nodes or other cells by its nearest neighbors within physical distance. And its node attributes are the individual proteins or markers that it expresses. We train this algorithm with this information, and then we can use this now algorithm to annotate additional new unlabeled data sets. And it's also able to recognize these novel cell types like I mentioned in the earlier algorithm. So if we have a labeled data set, for example, of our healthy human intestine, this is something of how it might look. Each individual data point is an individual cell, and the color represents the cell type within this section. If we leave this tissue section out and use our algorithm to predict, we can see that we get good alignment between the cell type prediction. And so we can use this algorithm. Once we have an annotated section from one of the donors, we can apply this to all the other donors and get our accurate cell types across the intestine accurately. And so we did this across 8 donors. And here are the results summarized. We can see we can identify many individual cell types across many different compartments. We can see that there are meaningful differences as we move from the duodenum to the sigmoid colon. Particularly, I'm interested in immune cells, and what is one thing that's interesting is that there's such a decrease in the percent of CD8s within the intestine. And this makes sense from an immunological point of view, where there's different access to the microbiota. So you might expect something like this. But what was especially interesting was there's a lot of large variance here in the percent of CD8 cells within different donors in the small intestine. And so we looked at what could explain this within the metadata. And one of the things that we saw was that people who had a history of hypertension had much fewer CD8 cells within the small intestine than people who did not have a history. And this was interesting, but also surprising. And so I also looked at endothelial cells, and there was a decrease also in endothelial cells, which you might expect. And so we tried to figure out where these CD8 cells were lost, whether it was broadly across the intestine or a specific area, since we had the location of this from the multiplex imaging. And what we found was that these were specifically CD8 T cells that were lost from the intraepithelial T cells that sit between the epithelial cells in the mucosal area. So this is important implications in autoimmunity, chronic inflammation, and also cancer. So, this is something else to follow up on. Another thing that we found correlated with metadata was that this inflammatory macrophage, M1 macrophages, correlated very strongly the percent of these macrophages with the body mass index of individuals. And it's known that the increases in body mass index are associated with a greater propensity for gastrointestinal disease. While our donors did not have any gastrointestinal disease, this could be a predisposing factor or a pre-indicator that this gastrointestinal disease is happening. We looked where these M1 macrophages were. These were primarily focused within the mucosal area of the intestine, while their M2 non-inflammatory counterparts were spread throughout the intestine. And this has prompted a lot of interest, and will be the subject of my third project that I'll discuss later. But so far what I've kind of told you is we've looked at breaking down a complex thing like tissues into their individual pieces. And I've showed some data about how we analyze those pieces and where they're found or not found. But we also want to understand how individual pieces fit together to form modules. And even beyond that, how do modules fit together to create multi-modules? And so one of the ways we do that in a computational way is to look at the neighbors around each individual cell. And once we see what individual neighbors are around each individual cell, we count them up, we put them into a count vector, and then we use unsupervised clustering to cluster these into also specific clusters that represent a cellular neighborhood or a distinct composition of cell types, which we call cell neighborhoods. And so what does this look like for our intestine data? Here we have the fluorescent imaging, and here we have the cell types that have been identified from this data and colored in data points. And then if we do these cell neighborhood analysis, we can start to see that we're identifying major structures like an inner and outer follicle region that are known to be cellularly composed differently. And vasculature shown in yellow, stromal areas in gray. And what does this look like computationally? Well, we can look at these clusters and see what cell types are enriched or depleted. And for example, this plasma cell enriched community or neighborhood we can see that obviously plasma cells are enriched, but also CD4 cells, dendritic cells, macrophages, and depleted in other cell types like epithelial cells. And we can apply these, that allows us to learn what the neighborhood is or what its function is, and we can name it, and we can apply these neighborhoods across tissues and across all of our donors to learn not only how cell types are changing, but how cellular neighborhoods are changing as we move across the intestine. One of the things that I find interesting, though, is if we think about vasculature here in the submucosa versus the vasculature here near the follicle versus the vasculature here in the mucosa, these are in very different environments. So even though these are a multicellular unit, we can find these multicellular units in different contexts. So how do we think about that spatially? And we have a lot to kind of draw from in the geographic sense, where we already think about this at multiple scales. So we think about neighborhoods that form counties, counties that then form states, and then states that then form countries. And it's kind of a similar approach that we took to the intestinal data set. So we first identified all the individual cell types in these images. Then we identified the cellular neighborhoods and how each individual cell type is connected to these different neighborhoods. Then we applied this concept again and went to neighborhoods of neighborhoods, which we called then communities. And these communities are larger spatial domains that are composed of conserved neighborhood arrangements. And then we can go even further to start to define tissue units. And we can see at each level, and we can also connect proteins to here, how each is connected across the different scales. And we can learn a lot from a graph or analysis like this. One of the things that we found and was really interesting was there was this one community that was the intersection of multiple immune and epithelial neighborhoods. And what we found was that, and why we named it adaptive immune enriched, was that it was at the base of the mucosa, which is near the stem cell crypt of the intestine. There was a conserved enrichment for CD4 and CD8 cells that wasn't previously appreciated. So this discovery of this, but also that we use then these communities, because you can see that they're spatially layered, to identify where in the position of the mucosa we were, so that we could define different layers. And we were able to observe different immune compositions or layering that happens in addition to the epithelial layering that was known to already occur. So, we had these other single-cell RNA-seq and single-cell ATAC-seq data sets, and a talented PhD, Bokai, developed this algorithm called MaxFuse, which can merge multimodalities that are across modalities. So there are some linked features, but there are some things that are not shared between the different features. And this algorithm enables single-cell matching across these two in an accurate way because it uses all the data. And so this is what the data look like in the small intestine and colon, where we have, this is from the single-cell RNA-seq. Here in the same embedding space, we have the codex individual cells. And then next to that, colored separately, are individual cell types. And you can see that there's large agreement between the two assays where both are the same cell types are colored similarly. And if we look at an individual cell type like goblet cells that are in epithelial cells, and we look at proteins that they should express like MUC2 or RNA that they should express, we can see that across both assays, these expression are strongly related and relate to the overall definition of these cell types. So with this merged linked data, we thought, what can we do that we can't do separately in a single-cell RNA-seq or a single-cell ATAC analysis? And we said, we have these different neighborhoods from the intestinal data set. Why not take these neighborhood labels and understand gene expression differences between the different neighborhoods? So we took the cells from these different neighborhoods and did exactly that. We generate a lot of differentially expressed genes between each individual neighborhood and all other neighborhoods. And if we take an example such as outer follicle here and put it into a gene ontology enrichment, we can start to understand and see things that we would expect, like B cell receptor signaling, T cell activation, B cell proliferation, regulation of these events. So, just to conclude this first part, it's, I think, important to not only look at one scale of biology, but also when we're working with these spatial omics data sets, to think about our analyses also as multi-scale and not stop necessarily at one level arbitrarily. And by doing this, I think it will uncover more of these multi-scale relationships that are really important, like we saw at the stem cell niche of the intestine. And thinking about, this first integration of the data set, we used only minimally, and I think there's a lot more to learn there about how the intestine is regulated and organized and developing over time. So now I'm going to switch gears a little bit and talk about our use of this technology within disease. And specifically, this was a project that I was fortunate to work on with a group of amazing collaborators, in addition to the first, which was an amazing effort by many, many individuals. But here we were funded by Cancer Research UK Grand Challenge Award to understand this general concept of chronically inflamed tissues and how they generate cancer. And this happens in the esophagus, lungs, colon, and stomach. And the similar processes happen where you start with a normal epithelial, but then you have some sort of chronic inflammation. Then the epithelial adapts and changes its type, which is called metaplasia. And then it continues to progress towards cancer as it moves to dysplasia, where you get more proliferation, and then it starts to become cancerous and invade different environments. And we see this when we have chronic inflammation, the same sort of pattern happens. And so there's some thought of, okay, the stroma is really important to understanding these processes, not just the cancer cells themselves. And specifically, I'll show you data from esophageal cancer, which derives from Barrett's esophagus, which is someone who has chronic reflux disease of the acid hitting the esophageal wall. Some set of people will progress and develop metaplasia, which is this change in epithelial lining. And people will get extensive surveillance, because some will never progress, but some will progress to dysplasia. And at that point, there's a resection that happens, an ablation, and there's a subset that fail that and move to cancer. And there's a high failure rate once someone develops cancer. It's a very aggressive cancer. So we, with the team, acquired 3 major data sets. First was to characterize kind of an atlas of Barrett's esophagus across different disease states, from normal to metaplasia to dysplasia to cancer, from the same individual. The second one was to characterize differences between esophageal cancer that was derived from chronic inflammation versus of other causes. And then the third was to look and compare people who have progressed versus people who have not progressed in Barrett's esophagus, and see what their samples could tell us about the environment. In total, we have more than 40 donors, and we derive this from our Codex multiplex imaging of these tissues, and to look at these interactions across scale. So again, we'll start with some of the Codex imaging that we did. Here on the top, we have the fluorescent imaging with a subset of the markers. And here below, I'm only showing the epithelial cell subtypes, so not all the other immune cell and stromal cell types, since we identified 57 unique cell subtypes. Here we can see in the normal esophagus, it's primarily squamous epithelial, as we might expect. And this is also shown in the different cases that we are able to profile. But when we move to metaplasia, you can see the epithelial changes quite drastically and changes into a more intestinal-like state that is characterized here and also quantitatively below. As we move to dysplasia, the epithelial becomes more aggressive. It gains p53 and loses differentiation markers. And as we move towards cancer, we get even more loss of these cell type specific epithelial markers like a squamous or intestinal like epithelial, and it becomes more dedifferentiated. And so we describe these changes as a change in the epithelial cell state, where it goes from the correct state to a misdifferentiated state towards a dedifferentiated aggressive state. Then we looked at the microenvironment, and one of the first things that we noticed was that as you progress towards tumor, you have an increase in regulatory immune cells, which is interesting because also you have this, it's a kind of a disease of chronic inflammation. Another cell type that was really interesting was that we saw an increase in these CD36 high endothelial cells. And the reason that this is interesting, one, because we saw a negative correlation between these Tregs and endothelial cells in our samples, but also our collaborators had seen that change in CD36 within the stroma changes the response in breast cancer. So this seems to be a conserved tissue signature of stromal remodeling in cancer, and that impacts overall outcome. Additionally, with our collaborators, we saw in our progressor versus non-progressor data set an increase also in CD4 Tregs in progressors compared to non-progressors, indicating that this regulatory cell could be preventing a good immune response in these tumors and enabling progression. Another thing that we did after this was looking at cell-cell pair enrichments. And the way that we did this is we permuted all the cell type labels across the image and then looked at cell-cell interactions that happen more than chance. And so, for example, here we're seeing in metaplasia this endothelial CD36 high endothelial cell interacting with our foveolar or an intestinal-like epithelial cell more frequently than we would otherwise expect, and more frequently than in dysplasia and tumor samples. And if we look, we can also see this where we have the CD36 high endothelial cell interacting with these MUC5AC or foveolar epithelial cells. We can then do that to understand what's changing in metaplasia and what is also changing within dysplasia, and we can start to see how we might observe things like foveolar cells starting to interact with other cell types like plasma cells or lymphatics more in dysplasia, and continue to see these different trends, and including an enrichment for neutrophils interacting with different cell types within the tumor samples. And if we synthesize the different trends here and look at what's enriched in tumor versus other samples, we can start to see that there are certain cell-cell interactions that are expressed within the tumor, or not overexpressed, but found within the tumor. And there is a large node around these neutrophils where they're connected to epithelial cells and other antigen-presenting cells or stromal cells, suggesting an important role in continued development or aggressiveness of the tumor. So one of the other things that we found that was really interesting was, like I mentioned in our intestinal data sets, we found that one of the important cellular neighborhoods was plasma cells. And I didn't mention so much of these plasma cell neighborhoods, but they're found within this mucosal epithelial space, and they also play an important organizational role within the middle layers of the intestine and associate with transit amplifying cells. But not only did we find that there, we also found that these plasma cell neighborhoods within our tissues of metaplasia. And what's interesting about these plasma cells, recently there have been a lot of papers that have come out to suggest that plasma cells predict outcomes in immunotherapy, while immunotherapy was traditionally thought to be a T cell target. And so we looked within our non-progressor and progressor data sets for these plasma cell neighborhoods as well. And what we find is that there are higher amounts of these plasma cell neighborhoods in non-progressors as compared to progressors, further suggesting there's some protective role of having plasma cells within these chronically inflamed tissues. And, we went beyond looking at plasma cells themselves to look at neighborhoods more broadly. And so we created these neighborhood maps of images that I've shown you previously, where the cells are individual data points and the colors are the neighborhoods. And we can find neighborhoods that represent these tissues, just like we did with individual epithelial cell types. Like here we see mostly apical squamous, and then we transition to a specialized intestinal-like neighborhood. But what you can notice is that each sample is fairly heterogeneous and there's a lot of variance. And that's because even within one sample, you can find many different types of epithelium where you can find maybe 50% cancer, 10% glandular, and 40% even normal epithelial. And so what we did with these percentages of neighborhoods is that we plotted them against each other to see where they correlated. And we saw that we could very accurately, across all samples, regardless of their official diagnosis, correlate these cellular neighborhoods with the pathologist score. So we can look at normal squamous, or we can look at metaplasia and find these neighborhoods that associate. So one of the things our collaborators also found was that they looked at different pathologically defined basically glands that develop, or epithelial characteristics that develop in the course of this disease. And what they found was that the more diverse this ecosystem of glands, the greater risk for someone. And so we thought about, okay, well, this is done pathologically, but can we do this from the ground up with these cellular neighborhoods? And so we can think about taking these cellular neighborhoods out from each other and creating tissue graphs that we could permute, and then think about which cellular neighborhoods are interacting more with each other. And we did this concept and applied this concept of multi-scale analysis of tissues in this data set as well, where we looked at not only the neighborhoods, but we also looked at these communities, or these neighborhoods of neighborhoods, which are these larger scale organizational patterns that we see in the tissues. And one of the things that was interesting is that there was multiple neighborhoods that correlated with invasive cancer, but there's only one community that correlated with invasive cancer as diagnosed by the pathologist. And this was this inflamed mature intestinal community. And what we saw from this was something that we measured was Shannon's diversity, which is an ecological principle where you measure the number and percent of each species to see how diverse it is. And what we found was that this normal community, the squamous epithelial, which is what originally normal esophageal epithelial is, was the least diverse. And this inflamed mature intestinal community was the most diverse out of all the communities. And what this suggests is that, and this is also true of just looking at cell type proportions within our progressors and non-progressor data sets. And what this suggests is that we start from our normal epithelial and we progress to a misdifferentiation to intestinal epithelial, but also the stroma is misdifferentiating to become more intestinal-like with these plasma cell enriched neighborhoods, and then becomes more aggressive into dysplasia and cancer and more dedifferentiated. And also the stroma is becoming more dysregulated and dedifferentiated as well. And so this is, I think, a very interesting finding and suggests a lot of interesting follow-up perturbations and studies to learn what is causing what. So we then went from this to use the PhenoCycler from Akoya to identify some spatial transcriptomics, where we looked at individual molecules that we were interested that overlapped with our Codex multiplex imaging panel. we can see that we get good expression of the different markers in the different samples to understand more molecularly what was going behind this. And from this, we can also identify individual cell types and individual neighborhoods that might overlap with our codex-derived ones. And you can see that there are major differences between our pre-progressors and progressors. Specifically, we started to look at things that might be involved in rearrangement of the stroma, things like chemokines and extracellular matrix remodelers. And here we, just by plotting these ones in these images, you can get a sense that there's a much greater expression, some molecules in non-progressors and others in progressors. And because we can quantify this, we can also see that that we have expression in certain cell types that's greater in non-progressors versus progressors. So not only in the overall sample, but for example, this CCL28, which is a recruitment factor for B cells. And we can also look at things within certain neighborhoods. So we can look at within certain neighborhoods what are greater expressions than in others. And then finally, within fibroblasts, within neighborhoods, we can look at specific values. So we can get very detailed because of the spatial features and molecular features to understand what potentially could be causing these rearrangements and spatial reorganizations. So we could think about treating or targeting these in the future. So just to conclude this part, we integrated kind of a top-down and bottom-up approach where we're taking traditional pathology to guide what we're doing in these spatial omics studies, but also going back and forth between these to learn from each other. And what we've learned is that, these cancers could be a problem of the microenvironment just as much as they are a molecular problem. And so thinking about how do you reverse this with non-traditional cell type targets like plasma cells or fibroblasts may be important. And then, we see this structural dedifferentiation that mirrors the epithelial differentiated. And, it's a chicken and egg question of which came first. And then, I think we didn't get into this too much, but there's conflicting forces. There is incredible inflammation, but there's also incredible immunosuppression that's happening at the same time to allow the tumor to progress. And how tissue does this and what wins out, I think, are interesting questions to look for in the future. So I mentioned this, our macrophage-specific areas in the human intestine. And Magda, who is an amazing scientist at Stanford and will probably have her own lab soon, and whatever place gets her will be very lucky. She was interested in human macrophages, and she reached out because she had profiled all of the public single-cell RNA sequencing data sets on macrophages and defined many different types, more than just the M1 or M2 that I had previously discovered. And she had gone on to definitively identify cell type markers that could mark these different macrophages. And what she was seeing also matched what we were seeing, where there were specific subtypes in the intestine towards the top of the crypt versus the bottom. And so what kind of we were interested in was, and she also found them separated in colon cancer and where they were within the colon cancer. So some were mostly in the stroma and some were mostly in the areas where there was tumor nest. So what we did is we developed markers towards these individual macrophages, and we're able to identify basically macrophage niches within the colon, normal colon tissue and colon cancer, and link these back to Magda's original findings of how and what these cells are doing and what they're doing differently. So there's some that are involved in efferocytosis, others that are involved in plasma cell niche maintenance, others that recruit and interact with neutrophils, and others that are associated with necrosis and hypoxia. And all of these are spatially segregated from each other and likely accomplishing different things. And as we learn more about these different types of macrophages, and Magda has gone on to show, some are associated with response or not responsive therapy. These could be specifically targeted in a therapeutic approach, since they have different roles in the tumor microenvironment. And this is just a summary. You'd find more by reading the paper that was put out in Cancer Discovery earlier this year. And we also looked, in addition to normal colon and colon cancer, Magda also looked at normal breast and breast cancer. So, that was a very quick few-minute summary, but, I think it highlights how we can leverage public data sets to drive biology, both single-cell RNA-seq and some of these spatial data sets, how we can think about spatially resolved phenotypes of cells that might appear as noise otherwise. And I didn't mention this, but Magda's put together a nice multiplex imaging viewer system through tissueviewer.org if you want to try that out. And just to conclude overall, I mentioned our work in looking at the healthy human intestine, our work in understanding chronic inflammation-induced cancers, and also our work in looking at different tissue microenvironments for macrophages. But what I didn't tell you was that we also have a story thinking about how does metastasis work in head and neck cancer, and also work that we've done on evaluating how effective cell therapies change the tumor microenvironment compared to non-infective cell therapies. And in each of these cases, what's an interesting concept that comes up again and again is that cells are immune cells, which are not typically thought as structural cells, because they migrate, they go in and out of tissues, are really important for the overall organization of tissue, both within health and disease. And I think more appreciation for that will allow control and resolution in disease and medicine in the future. So just to thank everyone, a lot of collaborators, both within the Nolan Lab, where I did my postdoc, and outside of the Nolan Lab across all of these projects, and my funding through an American Cancer Society fellowship and through the NIH. And I'll just conclude to say that, I've started my lab in January at Duke in the biomedical engineering department, where we continue to use these technologies like the Akoya phenocycler fusion and use it to profile spatial organization of tissues across different scales. We develop computational approaches to understand these data sets and extract biological mechanisms. And then we use engineering approaches towards manipulating cells or tissues to understand how we can perturb and understand these even better. And with that, I'm happy to stick around and take some questions.
Excellent. Thanks so much, John, for the great presentation. All right. And with that, we'll just jump in with the first question, a good one here. John, how do you decide on what marker panel to use for a study?
Yeah, this is a good question. I think, 50 plus markers seems like a lot of markers, but quickly you run into, conflicting things that you might want to learn in a study. And in a lot of studies, especially if they're exploratory, we'll look at major cell types within all the tissues and figuring out what markers are necessary to identify those specific cell subtypes. And a lot of times having multiple markers on a cell subtype is necessary for efficient clustering downstream after cell segmentation. In other cases, it's driven a lot by the biological questions. Other times we don't care so much about knowing all the cell types, but maybe there's a few functional cell types where we really care about. So yeah, I think, you're balancing budget with, the biology you're interested in with how much previous knowledge going into it that you have.
Yeah, it makes a lot of sense. Thanks. All right. Next one here. So have you used this technology to understand different therapies?
Yeah, so another project that I didn't share here, we use this technology to look at T cell therapies within solid tumors and also within immunotherapy treated melanomas within metastatic melanoma patients. And I think those type of studies are really interesting. One, because in animal models you can do perturbations, but then also and you can also look at things dynamically, which we did. But also you understand from those perturbations direct consequences in the multicellular neighborhood. So one of the things we found was that by changing the cell phenotype of these T cells, when we give them to these in the solid cancer setting, we can drastically change how the T cells go in to different locations, but then not only go in and kill tumor, but also reorganize the tissue microenvironment. So yeah, definitely we apply that within therapeutic settings.
Fantastic. So somebody was saying that they don't see fibroblast types here. Is there a reason for that? There are some very interesting spatial differences in the GI tract for them, especially expressing different BMPs, etc.
Yeah, good question. So in the intestinal story that we did not have markers set up to identify subpopulations of fibroblasts. In general, we had a general marker, a couple of general markers, and labeled them, I think, as stromal cells, but we didn't go into specific subpopulations. And definitely they're of interest in intestinal biology, but we just didn't have the markers and for these samples. Yeah.
Excellent. Did you look at mast cells like MMC and CTMC?
Yeah, good. Another good cell type. So we did look at the ICC subtype within the intestine. Now we're continuing this work in the intestine, or I should say, we and the collaborators have continued it, and we've added in a few different fibroblast markers and then also mast cell markers into this new panel, as well as subpopulations of plasma cells. So we are getting greater resolution on some of these more interesting cell types that came up within our first study.
Excellent. So how do you plan to integrate spatial transcriptomics and codex?
Good question. Yeah, I think there's a number of different approaches. Again, I think it depends on the study and the biological question. In many studies, we're doing subsequent sections of codex and spatial transcriptomics and registering spatially and then integrating across that dimension. In other studies, we don't have those integrations, but they come from the same sample, and so we can start to apply things like MaxFuse, which I introduced here, which align single cell data sets across different modalities. Eventually, hopefully, we'll have some techniques where we can directly do both on the same samples, but I don't think those are prime time and ready to go. But yeah, I think that there's a lot of different ways that these can be incorporated. Sometimes, we also do them in different sequences. We might profile many tissues with codex, find interesting tissues or interesting sets of areas, and then profile them with spatial transcriptomics. And the opposite can also be true, where we profile one or two tissues with spatial transcriptomics, get a few hits, and then design a focused codex panel to then look at many different slides.
Excellent. All right, can you talk a bit about the advantages and limitations of spatial transcriptomics for your research?
So I would say I'm a fairly new lab, so I'm not as wealthy as some of the other established labs. And so because of that, there's a significant cost there that comes with spatial transcriptomics. It comes with a great wealth of data as well, but in doing many replicates across many slides is a little unfeasible for me. And so I would say that that's one of the bigger challenges. Otherwise, it's another great source of data. You have to have people that are experienced, I think. Also, for me, that's less of an issue since we do this kind of stuff all the time. But I think that's what I see when I'm starting to collaborate with a lot of people, is if you don't have the experience to know what to do and how to normalize the data and how to filter out bad data and what to trust versus not, I think that can be very tricky, not to mention all the other downstream like spatial analyses that you can or could do with the data.
Fantastic. Okay, were the Treg increases associated with any treatments in the groups, proton blockers or absence of proton blockers, etc.?
Good question. I don't know if I had that information. I'm sure our collaborators did, who were the clinical source of the samples, and that would be interesting to look at. The sample set that we looked at in the codex was small enough that if there was heterogeneity in treatments, like with proton blockers, I don't know if we would be able to pull that out, but we ran single-cell RNA sequencing on many more samples than we did actually the codex imaging, and so maybe that could be pulled out with that data. But yeah, interesting question.
Yeah, definitely. Have you looked at transcripts or proteins originating from endogenous viral elements and repeat elements generally?
No, I have not. Very interesting, though. Yeah, definitely would be very interesting to look at those. And I know that there are a few, colleagues of mine who are interested in along those lines.
Awesome. Maybe something to look out for. All right, next one here. A number of studies have documented the presence of immune cancer hybrid cells, particularly tumor cell macrophage fused cells in tissues and blood. Have you seen these, or will one be able to better discern these rare hybrid cells using spatial transcriptomics?
I haven't specifically pulled out these fusion cells. One, because I wasn't looking for them, and two, I didn't necessarily see them. One of the troubles with spatial omics in general, transcriptomics or proteomics, especially transcriptomics, is that segmentation is not 100% accurate. And because segmentation is not accurate, interpreting the single cell data as ground truth is you can't do it because there is errors in segmentation. And so you have to be very careful about the cell types that you call, looking at the raw data, going back and forth, because you could get, some protein signal from a neighboring cell into another cell, and you may think it's this fusion cell, but it really is just bad segmentation. That being said, the project with Magda saw that there were macrophages that were specifically eating up tumor cells. And so they looked like they were double positive in the data, and they really are double positive, but it's not necessarily there's some sort of fusion cell. They're actually like digesting or eating up, phagocytosing the cancer cells. So there is cancer cell membrane within them, and it's not noise and it's not segmentation error, but they weren't really this fusion cell. And I'm fairly naive about these fusion immune tumor cells. So that's what I can say about that. Yeah.
Yeah, great answer. All right, so in non-progressors, are there more TFH rather than TH17?
Good question. Something we haven't looked at, would also be very interesting to look at. Yeah, the biology there would be really interesting. Yeah.
Yeah, another one here. Are you seeing a tertiary lymphoid organization here with the B cells? They say hasn't there been shown to be an indicator or a better anti-tumor outcome?
Yeah, I think this question is referring to the Barrett's esophagus study. We don't see so much of a tertiary lymphoid structure like what I showed maybe in the intestine, that are these traditional structures with high density of B cells and then a lower density with CD4 cells and dendritic cells. But we do see these plasma cell communities show up, and follicles or like immune tertiary lymphoid structures have been shown to be an indicator of better outcomes in cancer. But yeah, these plasma cell neighborhoods are, maybe downstream of these tertiary lymphoid structures and what we saw to be more correlative with better outcomes. And we've also seen this in other tumors that it's not necessarily the tertiary lymphoid structures, but these plasma cell neighborhoods that seem to help people.
Excellent, great answer. And I think in the interest of time, we'll just have one last question, maybe a forward-looking question. John, what things in regards to the digestive system are you interested in the future?
Ooh, great question. Yeah, I think some of the things that I'm really interested in are how does the digestive system change with age, and does cellular organization also change with age, and can that be associated with things like senescence or chronic inflammation, and kind of tying together both the healthy tissue biology with diseased tissue biology. Yeah, I think these things I'm really interested in. But like the many questions that came up today, there's a lot of different avenues to explore.
Definitely, yeah, definitely a really a great field to keep your eye on. All right, well, John, thanks so much for the great presentation and your insights in the Q&A. It's really been a pleasure having you with us.
Cool, thank you for having me.
Yeah, our pleasure. Also, big thanks to the audience for joining us. And in closing, we hope you enjoyed this Scientist.com and Akoya Biosciences webinar, and we'll see you again next time. Have a great day, everyone.